Welcome, everyone, and thank you for standing by. Guests are in a listen-only mode until any of the question and answer sessions of today's conference. At those times, you may press star one on your touch-tone phone if you would like to ask a question. Today's event is being recorded. If you have any objections, of course, you may disconnect. And now I'd like to turn it over to your host, Ms. Catherine Hambleton. Thank you so very much, ma'am. You may begin.
Thank you, and good afternoon. I'm Catherine Hambleton from NASA's Office of Communications. Thank you for joining us today to talk with scientists and researchers about the studies they will be conducting aboard the International Space Station and in the National Lab. In about 1 week, SpaceX will launch its 22nd cargo mission to the International Space Station under NASA's Commercial Resupply Services contract. The Dragon spacecraft will carry more than 7,300 pounds of supplies and payloads, including materials to directly support dozens of the more than 250 science investigations and technology demonstrations that will occur during expeditions 65 and 66. Before we hear from each of our researchers, we'll have a brief science overview from NASA, and we will then introduce each speaker for a discussion on their research or hardware, including time for questions and answers after each speaker. We'll also have some time at the end for additional questions. Your phones are on mute now. To get into the question queue, you can press star one on your phone at any time. The operator will open and close your mic so you can ask your question. And we ask that you please stick to one question. If we have time, we'll allow reporters to ask a second question. About 1 hour after we conclude, you can listen to a replay of the teleconference by dialing toll-free 800-678-0756. That's 800-678-0756. First up is Jennifer Buckley, Deputy Chief Scientist for the International Space Station Program Science Office. Jennifer, I'll turn it over to you.
Thank you, Catherine. Good afternoon, and thank you for joining us today. It's our pleasure to share with you the science that will be launched on the SpaceX Dragon spacecraft on the afternoon of June 3rd. Before we hear from our researchers, I'd like to take a few moments to talk about this mission and ISS research. The International Space Station is an incredibly capable and versatile laboratory. Our focus areas of research include human research, biology and biotechnology, earth and sea science, physical science, technology, development and demonstration, as well as education. And over the course of the year, there are hundreds of experiments that are being performed on the space station. Across our international partnerships, nearly 3,000 investigations have been performed on ISS to date. That represents over 4,000 scientists worldwide and more than 100 countries globally. Research from these investigations will help us advance in areas critical to our future exploration programs, as well as contribute to improving life here on Earth. For this mission, the Dragon spacecraft accommodates a variety of scientific payloads within its pressurized capsule, as well as unpressurized payloads in the spacecraft trunk. This Dragon will carry nearly 7,300 pounds of cargo, with over 2,000 pounds of that as science payloads in the pressurized capsule. 3,000 pounds will be the ISS Rollout Solar Arrays, or iROSA, in the unpressurized Dragon trunk. When Dragon returns, it will then be bringing home roughly 4,200 pounds of cargo, the majority of that being science samples from the Human Research Program, biology and biotechnology, physical science investigations, education activities, and technology demonstrations. So as I mentioned previously, arriving to the space station in the unpressurized trunk of this Dragon will be 2 of the eventual 6 iROSA solar panels. These panels utilize the Rollout Solar Array technology that was tested aboard the space station back in 2017. These compact panels unroll like large blankets once in place atop of the current solar arrays. Combined with the 8 original arrays, when the full 6 of those iROSA panels have been installed, they will boost the power to station by up to 30% of our current capability. This allows us to maximize the use of our orbiting lab for many years to come. Similar technology will also be used on NASA's Gateway and DART missions, further demonstrating the space station's importance as a testbed for technologies to support Artemis and beyond. When the Dragon spacecraft arrives to ISS, it will be a busy time for ISS research on board. We're in the midst of our 2nd operational commercial crew mission, Crew 2, which is the first time that 2 astronauts from 2 different international partner agencies have flown in a commercial crew vehicle. We have Thomas Pesquet from the European Space Agency and Aki Hoshide from the Japanese Space Agency. There's a great complement of research from both JAXA and ESA and CNES, the French government space agency, aboard this Dragon, some of which you're going to hear about later in this call. Today, you'll hear from scientists and engineers behind just a handful of the research and technology demonstrations on this mission. There are also dozens more that span disciplines from human research to physical science and come to us from a variety of international and commercial partners. These investigations and all of the science hardware launching on SpaceX 22 are contributing to the advancement of human exploration of space, as well as benefiting our lives here on Earth. If you'd like to learn more about any of these experiments you hear about today or the other work we're doing on ISS, you can follow us on Twitter at ISS_research or download our app, Space Station Research Explorer, in the Apple and Google Play app stores. Thank you.
Thank you. Next, we'll hear from our researchers. As mentioned earlier, each speaker will provide brief remarks, and we'll open up the line for questions after each speaker. And again, to get into the question queue, please press *1 on your phone. So now we'll hear from Dr. Thomas Boosby, Assistant Professor of Molecular Biology at the University of Wyoming and Principal Investigator for the Cell Science 4 investigation. Thomas's laboratory at the University of Wyoming studies how organisms like the small but tough tardigrade are able to survive in extreme environments. Dr. Boosby?
Thanks very much for the introduction. So yeah, as stated, we're running the Cell Science 4 experiment. The title of that project is Using Water Bears to Identify Biological Countermeasures to Stress During Multi-Generational Spaceflight. So just to give a brief overview, spaceflight, and specifically prolonged spaceflight, can be extremely stressful for humans and other organisms. And this is primarily thought to be due to decreased gravity, so microgravity experienced during spaceflight, and increased radiation exposure. Understanding how to safeguard astronauts and other organisms against these stresses will be essential for ensuring a safe and productive long-term space presence and for prolonged missions. So to start to address this, we're going to be studying tardigrades and looking at their ability to cope with prolonged spaceflight on board the ISS. So for folks who aren't familiar with these organisms, tardigrades, or water bears as they're more commonly known, are a group of microscopic animals that are renowned for their ability to survive a number of extreme stresses. So some of the things that tardigrades can survive include being dried out, being frozen, being heated up past the boiling point of water. They can survive thousands of times as much radiation as we can. They can go for days or weeks with little or no oxygen. And importantly for this mission, they've been shown to survive and reproduce during spaceflight and can even survive prolonged exposure to the vacuum of outer space. So for Cell Science 4, what we're going to be doing is examining how tardigrades on the ISS cope with both short-term and long-term exposure to spaceflight conditions. So our animals will be sent up to the space station in a frozen, inanimate state. Once on orbit, they'll be thawed, and they will revive and be grown or cultured in a special bioculture system that was developed at NASA Ames. For our experiment, we'll have two sets of tardigrades. One will be grown for only 7 days, and this will allow us to get a snapshot of how those animals cope with sort of the immediate exposure to spaceflight. Our second set of animals will be grown generations on board the ISS. So for both these sets, both the short-term and long-term sets of organisms, we're going to be recovering those animals and looking at what genes they've turned on and off while on board the ISS to get an idea of how they're coping with short-term and long-term exposure to spaceflight on a level of gene expression. And ultimately, this information will give us insights into how one of the toughest organisms on Earth is able to survive the rigors of spaceflight. And our hope is that these insights will provide avenues for developing countermeasures or therapies that will help safeguard astronauts during prolonged space missions. And with that, if there's any questions, I'd be happy to address them.
Thank you, Dr. Boothby. We'll now go to reporters for questions. Please press *1 on your phone to be entered into the queue. And the operator will announce you and then open and close your mic to ask your question. Again, please stick to one question. Dr. Boothby, could you speak a little bit more about how studying tardigrades on the space station could help with the future of space travel and exploration and some of those avenues that you referenced at the end of your remarks?
Yeah, absolutely. So our main objective is to identify changes at the level of gene expression that are going on in these really tough, robust animals when they're exposed to the rigors of spaceflight. And so what we expect or anticipate seeing is that they will be upregulating, so turning on genes that help them cope with those stresses. And, you know, what we see in terms of how tardigrades are coping with these stresses, we think may be able to be translated to developing therapies for astronauts. So, for example, you know, one of the major stressors during spaceflight is increased exposure to radiation. This can generate a lot of reactive oxygen species, which are damaging chemical moieties that can be very detrimental to organisms in biology. And so, you know, for example, if we see that tardigrades are producing a lot of antioxidants or reactive oxygen species scavengers, which are basically molecules that help deal with these really harmful reactive oxygen species, you know, that might give us insights into how we could safeguard humans, for example, by supplementing their diet with with foods with increased levels of antioxidants, or other approaches such as that.
Thank you. We have a question from Amy Thompson of space.com.
Hi, thank you so much for taking my question. I was just wondering, how are you going to be able to measure how the tardigrades respond to different stressors and microgravity? And is that something that you're going to be able to repeat, I guess, with the other generations, since the tardigrades are going to be able to reproduce? Thank you.
So you were breaking up a little bit there, Amy, but I think your question was, how are we going to be able to see how they're responding to the different types of stressors? So specifically, how they're responding to microgravity versus radiation. Is that correct?
Yes, that's correct. Thank you.
Yeah. Okay. So for the animals on board, they will be exposed to all those stresses simultaneously. So from our space-flown animals, it will be difficult to deconvolve or to separate out which stress is causing which response. But in addition to these space-flown animals, we're going to be conducting experiments back here on Earth where we look at these different stressors separately. So we'll have animals here on Earth that are exposed to simulated microgravity as well as radiation, but then also organisms that are just exposed to microgravity or simulated microgravity, and animals that are just exposed to radiation. And so by sort of separating out these stresses in our ground controls, we hope to see which stress is contributing to which response in a more specific way. Thanks for your question.
Thank you. Next, we'll go to Dr. Jamie Foster. Dr. Foster is a professor in the Microbiology and Cell Science Department at the University of Florida and runs a research lab at the Space Life Science Lab near NASA's Kennedy Space Center. Dr. Foster is the principal investigator for the ADCEP Umami investigation, as she studies how spaceflight affects the normal, healthy interactions between animals and microbes. For her research, she uses the bobtail squid as a model organism for her research. Dr. Foster?
Thank you for the introduction, and it's great to talk with you all. Yes, I am the PI on the UMAMI mission, which stands for Understanding the Effects of Microgravity on Animal Microbe Interactions. And as was previously stated, I'm very interested in how microbes, healthy, beneficial microbes, are communicating with animal tissues in space. As astronauts go and explore space, they're taking with them a cacophony of different microbial species, and it's really important to understand how those microbes, collectively called the microbiome, is changing in the space environment and how those relationships are established. And because there are so many different species interfacing with astronauts, it makes it tough to really understand what one species is doing compared to another. So we've kind of started to use the bobtail squid as a model for a couple reasons. One is the squid has a very special organ called the light organ in its body, and that light organ is colonized by a single species of luminescent bacteria called Vibrio fischeri. And that bacterium colonizes that light organ, and the squid uses the light made by the bacteria as kind of an anti-predator defense. It allows the squid to kind of glow in the dark. But what's unique is it's just one species of bacterium and one host tissue. So you can really kind of follow the molecular dialogue that's happening as the kind of bacteria can initiate colonization. And the second thing that's important about the squid is they have an immune system called an innate immune system that is exactly or very similar to humans. And so we can take a lot of parallels of how the immune system is responding to these beneficial microbes in the space environment. So with this particular experiment, what we're doing is the squid are born without bacteria. They have to acquire those bacteria from the environment, just like humans do. And so we're going to launch 128 baby squid in the UMAMI experiment, and we are going to initiate the symbiosis. We're going to add the bacteria to the animals and watch what happens over the first few hours of this initial stages of colonization. And so we'll be preserving tissues over a timeline and then capturing kind of a molecular snapshot of what genes, kind of like the tardigrades, what genes are being turned on and off, and how is this molecular exchange happening between the squid and this beneficial symbiont. The symbiont is absolutely essential for the normal health of this particular animal. So as a control, we're also going to monitor animals that don't have the proper bacterium. They'll just be have water added to them. So we'll be able to have a control with the bacteria and a control without the bacteria to monitor the health of the animal in the presence and absence of their symbiont. And so we'll be able to also repeat this on the ground and just make sure that we can really tease apart the effects that the spaceflight environment or the microgravity environment has on this association. So this also, because it's answering questions about the immune system of animals, we think that there's going to be a lot of new insights. When you remove gravity from the scenario, there could be whole new pathways, whole new mechanisms of communication between microbes and animals that we might have never seen being here doing all these experiments on Earth. So this gives us a really unique perspective to look at questions of animal microbe communications in a really unique environment like the International Space Station. So that's kind of the gist of what the UMAMI experiment is all about.
Thank you. We'll now go to reporters for questions. Again, please press *1 on your phone to be entered into the queue. Dr. Foster, can you talk a little bit about the facility that will allow you to study these squid?
Absolutely. So the AdSet hardware is completely autonomous. It looks just like a box, but there'll be little bags of the paralarvae or the baby squid are only 3 millimeters, so they're very small. So we'll have replicate bags of seawater with the squid, and they're all housed within a little cassette. And the cassette has certain pumps that will add water when needed or add the bacteria when needed or pump out the water. And so everything is completely autonomous. so we don't have to take a lot of astronaut time. They'll just, as the experiment ends, they'll take the samples and preserve the tissues for return to Earth. But everything will be contained in these little cassettes and automatically run by TechShot's ADSEP hardware. So it'll make the experiment very easy and completely autonomous.
Thank you. Next, we'll have Jonathan Himmelfarb, Principal Investigator for the Kidney Cells 2 tissue chip experiment. Dr. Himmelfarb is Professor of Medicine in the Division of Nephrology at the University of Washington School of Medicine and a kidney disease specialist at UW-Medicine. This investigation could help develop better pharmaceuticals and therapies for treating kidney disease on Earth. Dr. Himmelfarb, please tell us more.
Well, thanks very much for the opportunity to talk about our work. So here on Earth, we have developed what's called a human kidney on a chip, or human kidney microphysiological system. And what we do is we take human kidney cells, and in a space about 1,000th the size of a drop of water, we're able to bioengineer the components of the human kidney. And for this project, we're focusing on what are called the tubules, which are the major part of the kidney that's involved in the excretion or elimination of sodium, potassium, calcium, and keeps the body in balance, acid-base balance. So the kidney tubules are very important to maintain human health. And on Earth, we have used this human kidney on a chip to study a variety of different kidney diseases and come up with potential new therapies for a number of diseases that affect the public health. But one kidney condition that we've been very interested in that's very hard to model with this system on Earth is what's called nephrolithiasis, or kidney stones. And kidney stones are formed when some of these minerals in these tubules, in particular calcium and oxalate as the most common minerals involved, form a crystal, and that becomes what's called a nidus for a kidney stone to develop. And one of the reasons this has been difficult for us to study on Earth is that the gravity, if we infuse these crystals into our system, gravity makes them drop down to the bottom, so they don't stay in suspension in the liquid as they would in the human kidney. So we've been really unable to model kidney stones here on Earth, but we think that the microgravity environment will allow us to effectively model how kidney stones formed, and importantly, what can be done to prevent kidney stones from forming. Now, kidney stones are a big problem for astronauts. Astronauts are at increased risk of developing kidney stones, and that can be mission critical if somebody develops a stone that requires surgery or is obstructing. But it's also a very common problem here on Earth. So about 1 in 10 to 1 in 11 adults in the United States will get a kidney stone, males more than females, and the incidence of kidney stones is increasing over time. And we have preventative therapies like drinking more water, but our treatments for people who develop multiple kidney stones or are at high risk for kidney stones are suboptimal right now. And so we're hopeful that these experiments that we're going to conduct on the International Space Station will be relevant to astronaut health and prevention of kidney stones in space, but also relevant to population health here on Earth and here in the United States. So the experiments that we'll be doing on the International Space Station, we will take these kidney chips that we make with human cells, and the size of the device that they're in is about a credit card size. And we will put 24 of these chips that come from diverse donors, diverse in terms of race, ethnicity, and sex, onto the International Space Station. We'll let them, we have a closed system that's very easy to run with its own pumps to keep fluids moving through the cells and keep the cells alive and functional. And after about 7 days of orbit, during which time the cells will adjust, the human kidney cells will adjust to microgravity, at that point in time, we'll have 3 experimental conditions. The first will be to infuse calcium oxalate microcrystals, very tiny microcrystals that are how we believe ultimately human kidney stones that cause problems form around these microcrystals. In a second set of these chips, we'll infuse the same microcrystals but with a fluid that we think may prevent crystallization. And it's a fluid that relates to dietary intake, so it would allow for a modification of diet in people at high risk for kidney stones. And the third condition will be no crystals at all, a control, or if it were a human trial, we will call that the placebo arm of the study. And these, we'll infuse these, and we will collect all the fluids that flow through these bioengineered human kidney tubules, and we'll also study the genes that are activated in response to injury and repair. And all these samples will be collected and then stored and then analyzed when the kidney chips come back from the International Space Station onto Earth. So we're very hopeful that this set of experiments will inform and elucidate problems that astronauts face, medical problems, but also teach us aspects of the biology of how kidney stones form and how they potentially could be prevented that would be highly useful for human health here on Earth as well. And I'll stop here and be happy to take any questions.
Thank you. We'll now go to reporters for questions. Again, that's *1 on your phone to be entered into the queue. Dr. Himmelfarb, can you talk a little bit about what you learned from the Kidney Cells 01 experiment and how that has informed this one?
Yeah, thanks for that, and that's really complementary. So this is the second launch of our human kidney on a chip. In a prior launch, we were also investigating in a different way why astronauts tend to get more kidney stones than other people. And part of the reason for that is that in the absence of gravity, the minerals, the calcium and the phosphorus and other minerals that are normally in bone, gravity helps maintain bone function. And so bones tend to demineralize in space. And one of the problems that astronauts get, in addition to kidney stones, is accelerated rates of osteoporosis or thinning or weakening of the bones due to the loss of the calcium and the phosphate from the bones. Well, where does that calcium and phosphate go? It goes to the kidney. And when it goes to the kidney, the concentrations of these minerals is increased in the tubules, and that tends to cause or precipitate and cause kidney stones, which is one of the big reasons why astronauts are so susceptible to kidney stones. It's also important that the kidney, these same cells, these same proximal tubule cells in the human kidney are the cells that make most of the body's activated vitamin D. And we know that vitamin D can be protective against osteoporosis. So we reasoned that if we could study whether these proximal tubule cells are making the right form of vitamin D that protects the body, that would be helpful. And if not, then astronauts could be supplemented with the activated forms of vitamin D. So our first set of experiments were examining how these proximal tubule cells in a microgravity environment make or don't make adequate amounts of vitamin D, the activated form of vitamin D. And what was reassuring is what we found out that these proximal tubule cells, even in a microgravity environment, can make appropriate amounts of vitamin D. We also learned that in this microgravity environment, and we know that space is an environment for a variety of reasons where there is accelerated aging, we also learned that these kidney cells were subjected to increased stress, and as a consequence of that, they make more inflammatory molecules, which could have an impact on human health. So in many ways, that first launch of the human kidney on a chip onto the International Space Station was highly complementary to the biology and the questions that we're asking here on this launch, because both center around the role of the kidney in, first of all, maintaining human health in space, and second of all, the susceptibility of the human kidney in space to kidney stones or nephrolithiasis.
Thank you. All right. Next, we'll speak with Kadambari Suri, Integration Manager for the Butterfly IQ technology demonstration. Kadambari is an integration engineer at NASA working on multiple projects related to advancements in the medical field for human space exploration, particularly in support of NASA's Artemis missions to explore the moon. Kadambari, please tell us more.
Well, thank you for that warm welcome, as well as the opportunity to come and speak to you all about our project. The Butterfly IQ demonstrates the effectiveness of a portable, commercial off-the-shelf ultrasound device when used in conjunction with a mobile computing device such as an iPhone. Now, given that the IQ has an incredibly small footprint when it comes to mass, volume, power requirements, thanks to its implementation of ultrasound on a chip, a very simple and sleek design, a user-friendly mobile application, as well as the allowance of both real-time and deferred connectivity, this technology can provide important medical capabilities while boosting our potential for autonomy for future exploration missions beyond low Earth orbit. Butterfly IQ represents the first mobile health technology to be evaluated in spaceflight. If we can learn to adapt mobile health to the spaceflight environment, we can leverage the convenience as well as the diagnostic power of these devices to increase our medical capability while simultaneously reducing the mass and volume of future medical systems. This reduces both the crew medical risk while allowing engineers to meet the extreme constraints imposed by exploration missions. The purpose of our technology demonstration is to allow the crew to perform one or more autonomous imaging procedures using the Butterfly IQ. We have picked imaging locations that not only include organs at various depths and of various sizes, but also ones that we've seen to be of great interest to both our medical and research community. Once we test the functionality of the device in a microgravity environment, along with the effectiveness of just-in-time instruction methods for autonomous hardware use, we will not only be able to recommend this device for operations, but the lessons learned will also assist us in broader mobile health technology advancement activities. With that, I'd like to open up the floor to any questions.
Thank you. We'll now go to reporters for questions. And that's *1 on your phone to be entered into the queue. Kadambari, can you talk a little bit more about how you define a successful technology demonstration for this project?
Sure. So our success criteria are based on both the hardware and application performance. So we'll be considering things like image acquisition, display and storage of images, as well as functionality of the probe when using various imaging presets and modes. So things like Doppler or B mode and M mode for those who might be more familiar with ultrasounds. And one of the biggest things that we will be taking into account device usability feedback from the crew. Of utmost importance to us is that the crew, likes the device and is able to use it and is able to perform well with it.
Thank you. Next, we'll go to Maurice Marnot. Mr. Marnot is a human spaceflight engineer at the French ISS Payload Development and Operations Center. Maurice is the Pilote project lead, one of the experiments developed by the French space agency CNES for the Alpha mission of European astronaut Thomas Pesquet. Maurice?
Yes, thank you, Catherine, for having me today to present pilot experiments. So Pilot is a technology demonstration sponsored by the French Space Agency and developed in the scope of the Alpha mission of European astronaut Thomas Pesquet. but we are also involving US crew members. For example, Megan McArthur is one of our first test subjects. So pilot experiment design is based on fundamental neuroscience research results on how the lack of gravity is impacting what we call visual motor task performance. So visual motor task is basically using a visual information as an input to do an action or movement. And it can go from grasping an object near to you or to operate a telerobotic arm. So what we know is that astronauts have to relearn how to move around, and because gravity plays a key role on how the central nervous system consistently combines the various sensory information. And this reorganization is possible thanks to the brain plasticity. Knowing all that, the scientific team has designed specific techniques to assist astronauts while performing telerobotic tasks, and pilot experiment aims to validate them. So these techniques are based on using visual feedback and force feedback. For pilots, for the astronaut, pilot is really like a video game. So he or she would be wearing a virtual reality headset and is piloting a virtual spacecraft or a virtual robotic arm to do specific precision tasks, such as following a trajectory or reaching a target. So we have two different devices for the piloting. One is called the Space Mouse. It is a 6 degree of freedom joystick. It is the picture that has been used by NASA to illustrate this event. And then the second one is also a 6 degree freedom device, but on top of that, it can provide an active force feedback. So this force feedback would be used to help the astronaut piloting. If I oversimplify, it's like when your gamepad is buzzing when you are playing Mario Kart. So it really brings your attention that you need to take immediate action to keep your chance to win the race. So the project partner of this experiment is, of course, the European Space Agency and the French Space Agency. We have PI coming from the French National Center for Scientific Research, CNRS. We also have involvement of Tecnalia, which is a research center mainly located in the Basque Country in Spain. And we have also the support from the virtual reality company Oculus, which is part of Facebook, and that they provide us significant support to get our virtual reality headset qualified for use on board space station. So the expected benefits for pilots are to help to design the space-specific workstation for piloting spacecraft and teleoperated robotic arm. So this is really aiming for exploration. We also expect technology transfer to Earth applications such as telesurgery. And last but not least, we also hope that this experiment can get educational outreach, as the experiment is easily relatable for the kid and to the general public, as it really looks like a video game. It's some kind of serious game, if I may. And so actually the very first session on board ISS actually happened today with Megan and Thomas, but we will upgrade the virtual reality headset, which is flying with SpaceX 22 cargo, to enable higher performance, and it will be used for the later sessions. Thank you for your attention, and I'm ready to take any questions.
Thank you. We'll now go to reporters for questions. Again, that's *1 on your phone to be entered into the queue. Maurice, will you talk a little bit more about some of the specific challenges to make the virtual reality headset work in microgravity?
Yes, thank you. That's a very good question, because virtual reality headsets rely on gravity to get all the sensors bringing together their information. And without the gravity, you basically have to do specific development to still manage to get use of the accelerometers and gyroscopes that are inside the VR headset and maintaining the head tracking capabilities. So this is a huge challenge, and we make use of a parabolic flight campaign to be able to manage that and to fine-tune this development. One funny anecdote about that is that while we were in parabolic flight, actually the pilot of the plane was Thomas Pesquet.
All right, thank you. We do have a question from Marcia Dunn of the Associated Press.
Yes, hi. I'm sorry I joined rather late, and I was hoping to get a question to two of the earlier briefers, if I might. I was looking for a rough estimate on the number of tardigrades and bobtail squid that are going to be flying into space on this mission, please. And are all these specimens coming back live when the SpaceX Dragon comes back a month or so after liftoff?
I can answer. This is Joni Foster. I can answer about the squid. There'll be 128 of the squid larvae that are going to be heading up, and they will not be coming back live on that capsule. They'll be actually their tissues will be frozen, and they'll be returning on a later return. So they'll be stored for a while on the station.
And on the tardigrade side, we have 2,000 tardigrades approximately loaded into each one of our bioculture systems, or the cassettes for the bioculture system, and we should have 10 of those.
Great, thank you. All right, again, we can open up to questions for any of our speakers now. Please press *1 to be entered into the queue if you have questions. All right, that will conclude our question and answer portion for today. SpaceX is currently targeting 1:29 p.m. Thursday, June 3rd for the launch of its Dragon spacecraft on a Falcon 9 rocket from Launch Complex 39A at NASA's Kennedy Space Center in Florida. NASA will broadcast more about this mission live on NASA TV beginning Wednesday, June 2nd. You can listen to a replay of this teleconference in about an hour by dialing 800-678-0756. That's 800-678-0756. You can follow along with our researchers in these studies in orbit online at nasa.gov/station, and you can follow along on Twitter at ISS_research. Thank you all for joining us today, and stay tuned at also nasa.gov/SpaceX for more details about the launch and coverage on NASA TV, as well as an album of imagery that contains pictures of some of the science discussed today, such as the water bears, squid, ASEP, etc. And with that, thank you for joining us.